The overall goal of this aggregation assay is to assess the binding of Notch receptor with Trans-Ligands and its inhibition by Cis-Ligands.
This method can help answer key question how a genetic or pharmacological factor affects the binding of Notch receptor to its ligand and to alter the signaling. The main advantage of this technique is that it allows to evaluate the trans and cis binding of Notch receptor with its ligand distinctly without involving the overlapping effect of endogenous ligand.
Begin the assessment by spinning down signal receiving cells and resuspending them in Schneider's medium. Count the cells manually using a hemocytometer. Plate 5 × 10⁵ S2 and stable S2 Notch cells in each well of a 6-well plate in 1 ml per well of Schneider's medium. Next, add 7.5 micrograms of double-stranded RNA to each well and incubate the cells at 25 degrees Celsius for 24 hours. Add 0.7 millimolar of copper sulfate to induce the expression of Notch and incubate the cells at 25 degrees Celsius for 3 days. After incubation, count the signal-sending cells manually using a hemocytometer and plate 5 × 10⁶ stable cells in each well of a 6-well plate in 1 ml per well of supplemented Schneider's medium. Then add 0.7 millimolar of copper sulfate to induce the expression of ligands and incubate the cells at 25 degrees Celsius for 3 hours. Harvest the double-stranded RNA-treated S2, control, and S2 Notch cells by gentle pipetting, and plate 2.5 × 10⁵ cells per well in a 24-well plate.